Hi everyone, welcome to my Rakesh Sir Lectures channel. So today we are going to see about glycolysis. So as you know, it is one of the metabolic pathway, right, of our body. So before really going to discuss about glycolysis, I want to explain few things which will give better understanding about glycolysis. So first things you need to know about anaerobic fermentations, you should know about anaerobic respirations, and you should know about aerobic respirations. So if you see what is the actual difference between these two, between these three, then you will find that here to get the energy, the metabolic pathway of anaerobic fermentation and the metabolic pathway of anaerobic respirations and the metabolic pathway of aerobic respirations. Out of this all three, this aerobic respirations is the most advanced respirations or most advanced pathway to generate energy, right? So so what we can see here is, what is the common point in this all three is this the metabolic pathway for all these three is it starts with glycolysis. It starts with glycolysis, and so this is the glycolysis. So it starts with, that means it starts with glucose and ends up with pyruvate, right? Glucose and pyruvate with the series of reactions, with a series of biochemical reactions, the end product is pyruvate, right? Here also same things for the, it is a metabolic pathway, anaerobic respiration. So it starts with glucose, and with series of reactions you will end up with pyruvate, right? Here also first pathway is common. So it starts with glucose and end up with pyruvate. No, but what is the major difference? As I told, this there are three terms, and glycolysis occurs in the both all the three terms. So what is the major difference here is, first difference is anaerobic fermentation is first primitive. When you see yeast, when the fermentation occurs, the microbes and all, the anaerobic fermentation occurs. The more advanced or more evolved than anaerobic fermentations, when you have lack of oxygens or lack of mitochondria, then you will have anaerobic respiration. Okay, but when the most evolved or most advanced metabolic pathway is aerobic respirations. So in this, when you have mitochondria and when you have oxygens, then the pathway, the aerobic respiration will follow the some different pathway. So first step in all these three remains the same, right? So what is second step? Here in aerobic fermentations, there is no TCA cycle. You know, this after pyruvate, it will the cycle will go into the TCA cycle, or we'll call it as citric acid cycle, right? Or it is discovered by Krebs, so it is also known as Krebs cycles. So this is the Krebs cycles, right? It is a tricytric acid cycle. So this Krebs cycles is absent in anaerobic fermentation. And you know, again after this Krebs cycles, this molecules has to pass through the electron transport chain, right? ETC, electron transport chain. And when the electrons released from this TCA cycles and from glycolysis, that has to pass through electron transport chain, and then here what is in our actual ATP is released. So ETC is also absent in electron transport chain, is absent in anaerobic fermentation. So that means it is a primitive metabolic pathway, right? And that means only glycolysis is the source of energy productions. And you know, glycolysis will produce only 2 ATP, right? 2 ATP. That means only few amount of energy is produced in anaerobic fermentations. So during the evolutions, the metabolic pathway developed from anaerobic fermentation to anaerobic respirations. So let's discuss now what is anaerobic respirations. So now in this pyruvate, in the presence of mitochondria, what will happen here is it will runs to the TCA cycle, Krebs cycle, right? So this is the Krebs cycle, or it is also called as TCA cycle, and the electron which is released from glycolysis pathway and the electron which is released from the TCA cycles, all the electron will be passed through electron transport chain, right? And this chain, when the electron is passed through this chain, here what will happen? Energy is produced. That means ATP is produced. But you have to understand here is, in anaerobic respirations, I am not talking about anaerobic glycolysis. I am talking about anaerobic respirations. So in anaerobic respirations, glucose first converted into pyruvate, and from pyruvate it runs into the TCA cycles, and from here the electron whatever produced in the glycolysis and the TCA cycles, it will pass through the electron transport chain. So in this, even it is passed through the electron transport chain, but the last recipient is not oxygens. You are getting it? Here there is no ETC only. Okay. So here, even though the last recipient of this electron should be oxygen. Let me discuss first this, then I will compare with this. So both, once we compare, then we can compare and contrast what is the exact difference. So what is happening in the ETC is, first let's from pyruvate in the aerobic respirations in humans, and all of us will most of the time will have aerobic respiration, right? Unless we are not in high exercise and all. So in pyruvate, this pyruvate converts into acetyl-CoA, and then it enters into mitochondria, and this mitochondria will have TCA cycles, and this TCA cycle, right? And this TCA cycles will release some amount of NAD plus, that is protons, or you can tell as electrons. So the electrons released from these two glycolysis and TCA cycles, it will pass through this electron transport chain, that is chain of electron. So when this electron is passed through this chain, the last receiver of this electron will be oxygen, right? O2, oxygen. So once the last receiver is oxygen here, but in this anaerobic respirations, TCA will be there, this will be present. Here also TCA is there, TCA is present. ETC is present in anaerobic respirations. Here also ETC is present. Now what you have to see here is, here the last receptor of electron is not oxygen. The last receptor, maybe it is nitrate, or maybe it is sulfate, okay, or maybe it is phosphate. So these all things you can find. But here the last receptor is oxygen. Last receptor of electron is oxygen. So I have seen most student confused with anaerobic respiration with aerobic respirations. They have misunderstood that TCA and ETC is absent in aerobic respirations, but it will be present in aerobic respiration. But it's not true. It's not true, because in anaerobic respirations you find TCA and ETC is present. Here also TCA and ETC is present. But what is the major difference is the final receptor of electron here is not oxygen. Okay, the final receptor is not oxygen, but here the final receptor is oxygen. So based on the final receptors, this term has been given, anaerobic and aerobic. And this is the most developed and metabolic pathway. When we are going in detail about this all pathway, so we have a series of lectures on this. So first we'll go with glycolysis, then we'll go with TCA cycles, then we'll go with electron transport chain systems, and then we'll see how the energy is produced, and that means how the ATP is produced. So I hope you have a clear concept about this all three metabolic pathway. This is from primitive organisms like yeast till mammals like we have human bodies, like most advanced species. So what is the major difference? Here it is TCA is absent, here TCA is present, here also TCA is present. Glycolysis is present in all the species. So it is a primitive metabolic pathway, you can call it. Glycolysis is considered as primitive and most important metabolic pathway. So TCA is absent, here TCA is present, TCA cycle is present. Here electron transport chain is absent, here electron transport chain is present, here electron transport chain is present, right? I can write it here, ETC. Now already this is absent, so no need to talk about this. Here, even though electron transport chain is present, the final receptor of electron is not oxygen, rather it has nitrate and sulfate, right, or some other polar compound. But here the final receptor is oxygen. So this is one more major difference which differentiate them anaerobic and aerobic respirations, right? Even though TCA and ETC is present in both the pathway, but based on the final receptors, the name has been changed. Okay, the name has been changed. So now, once you know about glycolysis, is the primitive pathway, primitive metabolic pathway. So we are going to see about what is glycolysis, right, and how this pathway occurs and what are the different steps involved in this glycolysis, right? So what I can do is I will erase this and I will go with this glycolysis pathway separately. So what you can do here now is you have to write about or you have to know about glycolysis, and when we are discussing about glycolysis, you have to know it is the universal metabolic pathway which will be found in each and every organism, right? So one question is, in which cell of our body glycolysis occurs? Yes, tell me. In which cell glycolysis will occur? Yes. No. So glycolysis, glycolysis occurs not in a single cell, it occurs in each and every part of our cell. So every cells will get energy. First step is either glycolysis should be there, minimum glycolysis should be there. So glycolysis occurs in each and every cell of our body. Second question is, what we need to start glycolysis? What we need to start glycolysis? See, glycolysis, if you want to define it, it is one of the primitive metabolic pathway, right? Or it is one of the metabolic pathway of our body. So which converts glucose into pyruvate, right? So glycolysis is one of the metabolic pathway, or it is a metabolic pathway which converts glucose into pyruvate. Now, to start glycolysis, what we require? What is the first step? What is the first step we require to start glycolysis? The first step, of course, we require glucose. We need glucose, right? So first we need glucose, then only the energy can be produced. The glucose can be break down into smaller molecules, right? So all of you know like how the glucose will be glucose will be that means glycolysis should occur in cell, and cell need glucose. So how the cell receive the glucose? So you know about this. I can draw here as this is your food pipe. So here is your food pipe, here is your stomach, right? And this is your GI tract. So from here there is gastromucosal cell, right? Here is gastromucosal cells. So here will be some special kind of transporter for glucose or monosaccharides, whatever you will eat. Either it will, if it is carbohydrate, it should break into monosaccharides. So monosaccharides either fructose, glucose, or galactose. So this will get transported into liver, right? This will get transported into liver. So this liver now metabolize either fructose or galactose or glucose into glucose. That means if you are eating fructose also, maximum fructose will convert into the glucose, and then then glucose will be released into this from the liver to the systemic circulations. Let's assume this is your systemic circulations artery, or systemic circulation, circulatory artery. So systemic circulations. So even you are eating fructose, ultimately it is converted into glucose and it will release into the systemic circulations, right? Am I right? If you are eating galactose, or even it is breakdown into galactose monosaccharides, then it is ultimately it will pass through the liver before it reaches to the circulatory system. So galactose first it needs to converted into glucose, and then this glucose will be transported into the systemic circulations. And if you take glucose also, the glucose same things will be transported into the systemic circulation. So what you understood from this? Glucose is the primary source of energy, right? Glucose is the primary source of energy. So now from this systemic circulations, your cell need energy, right? So your cell need glucose. This is let's assume this is cell, right? So this is cell. So if these cells this is systemic capillaries or capillaries areas, and from this capillaries area, you know, the glucose is reaching here. So once this glucose reach here, the capillary membranes are permeable to glucose. That means this is cell wall, I mean the cell membrane, and this one is capillary membrane or blood vessel capillaries. So this glucose can easily diffuse into the space between cell and capillaries, and that is called as interstitial space. So my question is, what is the concentration of glucose in the interstitial fluid as compared to capillary? What do you think? Concentration in interstitial fluid of glucose will be more, or concentration inside the capillary it will be more, inside the lumen? What will be where the concentration will be more? Eventually the concentration of interstitial fluid glucose and concentration of capillary both are equal, because glucose are freely permeable to interstitial space. So glucose can easily diffuse or easily move into the interstitial space. So the concentration of glucose inside the capillaries and in the interstitial space are same. Okay, so okay, this is first things. Now tell me the second things. When you take this glucose concentration in the interstitial space and glucose concentration inside the cell, intracellular, so which one? Extracellular will be concentration is more, or intracellular concentration is more? there is no phenolphthalein, so extracellular is more as compared to intracellular, because cell needs glucose, and it's always utilizing the whatever the glucose is left out. So concentration inside the cells will be, concentration of glucose inside the cell will be less or low as compared to concentration of glucose outside the cell. Okay, now we have to see how this glucose from here it will move into the inside the cell, because this cell membrane is not easily permeable, or it is not permeable to glucose. So glucose cannot easily diffuse inside the cells, right? So this capillary membrane is easily permeable. So from here the glucose capillary lumen to interstitial space is easily it can diffuse, but the cell membrane is not easily permeable. So that means glucose cannot move directly into the cells, right? So that means it requires some transporter. It requires some transporter, and the transporters is known as this glucose transporter. We call it as glucose, the special type of protein, right? The special type of protein will be present, and that protein will be present in the membrane, and that membrane, just assume like this is one membrane like this. Okay, so from what it will do is that one glucose molecule will be sitting here, right? And once the glucose, it is loaded with glucose, it will just flip in, right? It will flip in. So this type of protein, this is called transmembrane, right? Transmembrane protein, right? Transmembrane protein. So it is like it is opening like this, right? So it is opening like this. So once it is opening like this, once the this protein, once it is loaded with glucose, right? Once it is loaded with glucose, then it will open like this inside, right? So glucose will be moved toward inside the cells, right? So this is the way, but it is taking the help of transporter. This is protein is a transporter. And other thing is glucose is moving down to concentration gradient, that is moving from high concentration to low concentration. So when any substance moves from high concentration to low concentration, we will call it as diffusion. So of course this is diffusion, but this diffusion is facilitated by a protein. So this special type of this kind of diffusions is called as facilitated diffusion. Okay, so actually there are two ways how this glucose will enter inside the cell. One is through the facilitated diffusions. I can put here like there are two ways how this should move. Okay, one is through this facilitated, I'll write here better. So one is through facilitated diffusion, and other is through sodium glucose cotransporter. Sodium glucose cotransporter. Mainly in kidney, in PCT, you have that epithelial cells, right? And that epithelial cells, the lumen membrane, they have sodium potassium cotransporter. That means when sodium and potassium will be loaded, then that again it will get flipped inside this. So from there sodium and glucose from this nephron lumen will be carried inside the cells, right? So this is in nephron or in kidney, you can see that in easily this sodium potassium cotransporter, both will be transported, right? So most of the time we'll see this facilitated diffusion. So these are the two way how glucose should move inside the cells. Now based on this transporter, what we will call this transporter? Glucose transporter, or in short we can call it as GLUT. Glucose, we call it as glucose, GLUT, glucose transporter. Okay, GLUT, glucose transporter. Such type of protein is known as GLUT. So till now, approx 14 kind of glucose transporter has been identified. Okay, anyway, we will not go in more detail of doubt. But obviously we need to mention few types of this glucose transporter, and it is easy for us to understand how this works. So only just by hearing the glycolysis chemical reaction, it won't works, right? So we need to know the mechanisms, how it works, how our body works. So this is very important. Understanding the concept is very important, right? So so let me discuss few things about glucose transporter. So once we are discussing about glucose transporter, so there are 14 types, but any 4 to 5 types we will see, which is the major or popularly taught, or it is very useful. So first is GLUT1. Glucose transporter 1. So glucose transporter 1 mainly present in the you can tell, okay, nervous system, central nervous systems. So central nervous systems, our the nervous system cells are very active and energy dependent. So they need GLUT1. That means this type of receptors, or this type of transporter will present in the that cell, nervous cells, central nervous system cell, and this GLUT1, that is one way. This is one-way receptor. That means glucose can enter inside the cells, but it cannot come out. Right? This is the one-way GLUT1, one-way transporter. That means if you just assume this is as neuron cell, so that means in these cells glucose can go inside, but it will not come out. So one way. So such type of transporter is known as which will only open in one direction inside the cells. So such type of transporter is known as GLUT1. Okay. Other type we have is GLUT2, and this is the special type of transporter. I hope you know GLUT2. So GLUT2 indicate, what does GLUT2? 2 indicate here 2 ways. I don't know, just yeah, of course. So this 2 indicates 2-way transporter. It is 2-way. So 2-way in the sense glucose can enter inside the cells, and when needed, it can this cell should release the glucose outside of the cells to the systemic circulations. Okay. So what do you think? Which kind of cells or which kind of tissue this GLUT2 should present? This is about CNS you can put, or nervous systems neuron you can put. CNS and neurons, or any neurons you can put. Okay. GLUT2. What do you think? Which kind of cells will require this GLUT2 receptor? Just think. See, GLUT2, that means this is both way. Cell when needed, they are getting glucose. When required, it is releasing the glucose. Right. So that means this cells or tissue has glucose storing capacity. So the most popular organ is cell is hepatic cells. Right. This is livers. So when you have high amount of food, high amount of carbohydrate, so what it will do? It will store the maximum glucose, and only the required amount is released into the circulations. But you fast for a long. You are not eating for a long period of time. What will happen then? The glucose level inside the systemic circulation will drop down. Again, to maintain that glucose level, what will happen? Gluconeogenesis will occur, right? And from this gluconeogenesis, what will happen? This, let me draw this. This is hepatic cells. So when you have here itself, I draw. This is hepatic cells. So when this is when you are taking food, so when you have high amount of carbohydrate, that means it is storing it, right? This hepatic cell is storing it, and then it is then that is a storing phase, and only few amount is released only few amount is released. So when you have high amount of food, the storing will be there, right? Glucose is entering. Glucose is entering inside this cell, right? If this is hepatic cell, glucose will enter inside the cell. But when you are in fasting stage, and when you are fasting stage, you know this systemic circulation blood glucose will start coming down. So once it start coming down, what this hepatic cell will do, this stored glucose again in the form of glycogen, right? So it will break down in the form in glucose, and it has capability again. The transporter will be present in the this membrane, and it helps this transporter to get that glucose from inside to out, right? Inside to out, and with the help of this, once it come out, then it will release into the systemic circulations, right? So this is the two-way. Can you tell me some other cells where the GLUT2 will be presented? GLUT2. That means you should know which organs or which cells will do gluconeogenesis. What is gluconeogenesis? What is gluconeogenesis? Gluconeogenesis. So, yes, tell me, what is gluconeogenesis? So, what itself is glucose? Neo means new, genesis means formation. Formation of glucose from non-carbohydrate source. The simple way of defining is some books they will write like, okay, formation of glucose from fats, right? But if you want to tell in simple term, it's not only fat. When the glucose formation is from protein also, that is also called as gluconeogenesis, right? So formation of glucose from either from fats or either from proteins, or a simple word you can tell it, from non-carbohydrate source, right? So formation of glucose from non-carbohydrate source is known as gluconeogenesis, and the most common organ which do this gluconeogenesis is liver, right? But there are some other organs also in our body, or some other hepatic, apart from hepatic cells, there are some other cells in our body which will do gluconeogenesis. Yes, and you know, again, our kidney, don't forget our kidney. We just remember it as a filtration organ, but it's more than that. So of course, again, this gluconeogenesis occurs in kidney, in renal systems, mainly in PCT, right? So that means it should occur in liver and kidney, because GLUT2 receptors will be present in such type of cells where there will be a storage of glucose will be there, or gluconeogenesis should occur, right? So these two, liver and kidney, are the most popular cells. Renal cells, nephrons, and hepatic cells are having GLUT2 receptors. When I'm talking about this, one more cell or organ is there which have GLUT2 receptors, and you know that it is very important, very important. That is very important. What is that is you know about, and the major magic of that cell is, let's assume this is that cell what I'm talking about. This is the cell, and the major magic of this cell is the concentration of glucose outside the cell. The concentration of glucose outside the cell, and the concentration of glucose, let's assume this is blood. Okay, blood and interstitial, both concentration is same, right? So I can just, and the concentration of glucose inside this cell. Okay, concentration of glucose inside this cell is equal. This is the one magic. Before we studied like, okay, and the concentration of capillary and this interstitial is same, that I can understand. Now this is one special type of cell in which this concentration of glucose in an interstitial fluid and the concentration of fluid inside the cell is same. And you know these cells. This is a pancreatic beta cells of pancreas. which has high tendency to or which is high sensitive to glucose. Even little change in the concentration can detect, right? Little and it can produce insulin based on that, right? It produce beta cells. That means this beta cell should be easily permeable, and it should have such type of transporter when this glucose level here is high, here also this glucose should move here, and the concentration here should also increase to maintain the equilibrium. Because whatever the blood concentration is there, same concentration should be here, so that in the same range, in the same degree, insulin should get produced, right? So this cell should be highly sensitive to glucose. Okay, so if let's assume you have eaten after 10 hours of your eating, that means once you are in fasting stage, so your blood glucose level will down. So one blood glucose level will down, so this also will get down. Once this will get down, insulin production will decrease. So this should have two-way transporter. This should have two-way transporter. That's why GLUT2 will be present also in beta cells of pancreas. Am I clear? I hope I'm clear. If anything is there, just put on the comment. I'll read the comment and I'll try to answer and give my best. Or else, if any doubt is there, based on that, I will create one more video so that it will be useful for you. Other GLUT3 glucose transporter. GLUT3. Now don't tell me like this is a three-way transporter. Just this is one way, this is two-way, but this is not the three-way. GLUT3 means it is a transporter. It is not most commonly used, but okay, fine, no problem. GLUT3, and other we have GLUT4. GLUT4 most commonly used. So GLUT4 is insulin dependent transporter. So insulin dependent, GLUT4 is insulin dependent transporter. Okay, that means whenever the insulin cell will open their based on this what I have discussed here, this one GLUT, this opening and closing. Once it is loaded with glucose, once the protein is loaded with glucose, or once the transporter is loaded with glucose, it should open down, right? So this opening and closing phenomenon will be facilitated by insulin. Okay, will be facilitated by insulin. So this is insulin dependent transporter, GLUT4. What about GLUT5? So GLUT5 is fructose transporter. It is fructose transporter. It is fructose transporter. So fructose transporter, as I told, fructose is absorbed from here. So once fructose is absorbed from here, once it reaches to hepatic system, most of the fructose will convert into glucose. But anyway, few amount of glucose will be released in the systemic circulations, and it will be which cells loves to utilize fructose. There is some cell in our body, and that cell loves to utilize fructose as their metabolic pathway. Out of glucose and fructose, which one is more sweet? Out of glucose and fructose, which one is more sweeter? Yeah, of course, fructose, right? Fructose is more sweeter than glucose. Now, this which part of your body loves to have sweet things? Which part of your body loves to have sweet things? That cells will love to have fructose, because fructose is sweeter, you know. So which part of your cells, or which cells? Taste bud, no? taste bud no. You people are thinking something else. So do you know about, you have a pair of organ, you know what testis, right? So testis will love to have fructose to run their metabolism, right? And you better know like how, what sweet things testis loves, right? Other is sperm cells. So both testis and sperm cells utilize fructose. And what sweet things they like? Okay, so now this is like fructose transporter, and this glucose 3 is facilitated, just simple facilitated diffusions, right? So based on that, okay, this is like, okay, kind of this glycolysis or glucose transporter. So once we know, but by this, with the help of this transporter and with the help of facilitated diffusions and sodium glucose co-transport, this glucose from interstitial space, it is reaching inside the cells. So now we are going to see in detail about once the cell reaches the once the glucose reaches inside the cell, how it works, right? Or how it metabolized, right? So what are the various pathway it will go off? Okay, so that we'll see in the next lectures. So just wind up the lecture.